Hello everyone and welcome back to my channel. In this video I will talk about the methods to estimate heritability. In my previous videos I have described the partitioning of phenotypic variance into different components. I have also already explained the definition of heritability and the types of heritability, which are broad sense heritability and narrow sense heritability, and I have also talked about the difference between the narrow sense and broad sense heritability. In my last video I have also discussed the misconceptions about the heritability. So if you didn't watch these videos, I would recommend you to watch them before watching this one to better understand the concepts of heritability. I have provided the link of those videos in the description of this video. So in this video, firstly I will again provide an overview of heritability, then I will discuss the methods to estimate broad sense heritability. Afterwards I will describe the methods to estimate narrow sense heritability, and at the end I will provide an example on estimation of broad sense heritability using twins data. So I will start this video by defining the heritability. Heritability can be defined as the proportion of the total phenotypic variance caused by the genotypic variance, and it is divided into two types, which are broad sense heritability and narrow sense heritability. Broad sense heritability can be defined as the proportion of the total phenotypic variance caused by the genotypic variance, whereas narrow sense heritability is defined as the proportion of the total phenotypic variance caused by only additive genetic variance. So there is only one difference between the broad sense and narrow sense heritability. The broad sense heritability is the ratio of phenotypic variance that is due to genotypic variance, and this genotypic variance contains all the three genetic effects, which are the additive genetic effects, dominance effect, and epistatic effect. But in case of narrow sense heritability, we only account for the additive genetic variance. So it is the ratio of phenotypic variance that is due to additive genetic variance. So what are the methods to estimate broad sense heritability? Actually, there is only one method to estimate broad sense heritability, that is monozygotic and dizygotic twin data. So broad sense heritability can only be estimated through the monozygotic and dizygotic twin data. I will explain this method in detail at the end of this video. So, and what are the methods to estimate narrow sense heritability? Actually, narrow sense heritability is mostly estimated using different statistical methods, which are regression, correlation, and ANOVA, or analysis of variance. So first we will see the methods to estimate heritability using regression. The first method is the offspring-parent regression. So this method is based on the regression of offspring on parent, and it is further divided into two categories. The first type of estimation of heritability through regression is the regression of offspring on one parent. If you have the data on offspring and only one parent, then you will use this method. But if you have data on both parents, male and female parent, as well as their offspring, then you will use the regression of offspring on mid parent. Mid parent is actually the average value of both the parents. The second method of heritability is through correlation between relatives. Correlation is the statistical method which is used to find the relationship between two variables. So we can use correlation between relatives to estimate heritability. So again we have two types of correlation between relatives. The first is the correlation between parent and offspring. I earlier said that you can use the regression to estimate the heritability, but you can also use the correlation between parent and offspring to estimate heritability. However, the most reliable estimate is considered the regression of offspring on parents. But still you can use the correlation between parent and offspring to estimate heritability. The second method is if you don't have the information on the parent and offspring and you have information of sibs, then you can use the correlation between sibs to estimate heritability. And these sibs might be the full sibs or half sibs. Full sibs are those individuals which have both parent in common. For example, the real brothers. Half sibs are those which have only one parent in common. So you can use the data on full sibs and half sibs, and you can find the correlation coefficient to estimate heritability. The third method to estimate heritability is the analysis of variance or ANOVA. It is also sometimes called as intraclass correlation. So intra-class correlation can be used to estimate the heritability, and it can also be further divided into two categories. The first type is intra-class correlation between full sibs. If you have data on full sibs, then you can find the intra-class correlation between full sibs to estimate heritability. And if you don't have the data on full sibs, you only have the data on half sibs, then you can use the intra-class correlation between half sibs to estimate heritability. So actually there are two methods to estimate heritability on the basis of relationship between full sibs or half sibs. First is the correlation coefficient, which I have already explained, and the second one is the intra-class correlation, and both of these methods can be used. However, the most reliable method is considered the intra-class correlation. So this method is most reliable. I will discuss these methods in very detail in next video. The fourth method to estimate heritability is the restricted maximum likelihood, also known as REML method. And this is a highly advanced method. So to estimate heritability using restricted maximum likelihood, you need different type of softwares which might be the Wombat, ASREML, SAS, Blup F90, or there are so many other software which can also be used to estimate heritability. And this method is highly reliable, and this type of method is mostly used nowadays to estimate heritability for different rates of livestock species. You can see different research articles where they use this type of method to estimate heritability. So now I will discuss the detail on methods to estimate broad sense heritability, which is monozygotic or and dizygotic twin data. Estimation of broad sense heritability on the basis of monozygotic and dizygotic twin data. Firstly, we will see what is the monozygotic twin and what is the dizygotic twin and what are the differences between monozygotic and dizygotic twins. So monozygotic twins are sometimes also called as identical twins. There are two names given to monozygotic twins. They can be called as monozygotic twins or can also be called as identical twins. And they develop from one zygote which splits and form two embryos, which means a single egg is fertilized with single sperm. A single egg is fertilized with single sperm, and then this fertilized egg splits to form two embryos. So that's why they are known as identical twins, because they are genetically same, because they are developed from only single zygote. The dizygotic twins are sometimes called as fraternal twins or non-identical twins, and in this case each twin develops from a separate egg. So fraternal twins are developed from the separate egg, and each egg is fertilized by its own sperm. So they are developed from two different eggs and two different sperms. That's why they are known as non-identical twins. So now I will provide an example on estimation of heritability using monozygotic and dizygotic twin data. As I have already explained that broad sense heritability can only be estimated using monozygotic and dizygotic twin data. So the formula to estimate broad sense heritability is the capital H square is equal to 2 into RMZ minus RDZ, where the RMZ is the correlation between the monozygotic twins and RDZ is the correlation between the dizygotic twins. So to estimate broad sense heritability, we need a correlation coefficient between the monozygotic twins for the same trait, and at the same time we also need the correlation coefficient between the dizygotic twins. This type of studies are mostly done on the humans, as they are not possible for different livestock species like cattle and buffalo because they don't produce twins. So this type of study is mostly done in humans. So to estimate broad sense heritability, first you will estimate the correlation coefficient between the monozygotes and the correlation coefficient between the dizygote. But here I am not going to do this example, as I have used the data which is already published and I have collected it from the Wikipedia. So this is the published data on the concordance between the twins, and in this example I will estimate the heritability of alcoholism in males. So this black bar, it shows the monozygotes, and this white bar, it shows the dizygotic twins, and this y-axis, it shows the correlation coefficient or concordance is the similarity between the twins, where one twin express the one trait and other also express the same trait, then this is known as concordance. And what you see here, 444 is the sample size, and for dizygotic twins the sample size was 653. So they used data on 444 monozygotic twins to estimate the correlation for alcoholism, and they used the data on 653 dizygotic twins to estimate the correlation for alcoholism. So the estimate of correlation coefficient for monozygotic twins was almost zero point 0.4, and the estimate of correlation coefficient for dizygotic twins was almost 0.20. So to estimate heritability, we need the formula, that is capital H square is equal to 2 into RMZ minus RDZ. We have the values of RMZ as 0.40, which is the correlation coefficient between the monozygotic twins, and we have the RDZ, that is the correlation coefficient between the dizygotic twins, and it is 0.20. So we will put these values in our formula and it will become like this. So now we have replaced these with the values. Okay, so we will solve this equation and our heritability would be 0.40. So the heritability in males for alcoholism is 0.40. So this was a video on methods to estimate heritability. I hope you enjoyed it, and if you like the video, please share it, and if you didn't subscribe to my channel, please do subscribe it.